through the content for Module 1 in biology, and we'll do it together as a group. And as we go through it, we'll do it as slowly as we can, but we've only got 2 hours. So if we feel we're okay with it, we'll move on. If we want to spend a little bit more time on it, we'll move on. So I'll go through a couple of slides. We'll stop and I'll say, okay, you can do the question at the bottom of that section on the Moodle, tick it off, and then I'll talk about the next page and so forth as we move through. By the end of the session, then you should have done first 2 topics in the Moodle. So I'm going to need to access the Moodle up screen as well. So you can see here, today we should do cell structure and cell function, about how cells coordinate. A number of people involved in the development of this Moodle. And I'd also like to qualify that we've done this in a very short space of time, in amongst, as you know, the busyness of teaching. So if we've made a mistake, or if you see an idea that might better explain some of the principles that we're trying to talk about, can you please tell us or share with us so we can make changes? It would be much better if we can fix it rather than leaving it in a situation that's not the best. Bottom there, next section at the bottom. So that then will take you into the next page of the booklet. This is written like a booklet, this Moodle. So each of these blue sections is a new page. When you go into each of these sections, there's a booklet within it with lots of pages. So you can see here we've got so this is Module 1. This is Module 2. 6 and 7 are Module 3. 10 Module 4. Click on cell structure. We're going to look at systems biology, at prokaryotic and eukaryotic cells. So on that first page there, you can see the content focus, and at the bottom, mind map. I'm going to give you a minute just to have a quick read through that before we go any further. Okay, so you've had a chance to look at the content focus now. What I'd like to draw your attention to is the fact that it says that cells are the basis of life. They coordinate activities to form colonial and multicellular organisms. Now, we don't do any colonial and multicellular organisms in this module. So I don't know why it's there. This module is only about cells. So that's very confusing. It also talks about tissue level, and again, we don't do it in this module. It comes up in the next one. And an incorrect word there says biochemical processes. It should say pathways. So it should be biochemical pathways through the application. It's a bit pedantic, but that's right. All right, so requirements for our pracs in our bio says 35 hours of course time. Which is fine. We're used to that. That's okay. What is different are what is the definition of prac. So in our last syllabus, we had a whole page of practical activities, graphing and drawing tables and analysing images. That was all practical. The only thing that now it says are laboratory experiments, including appropriate use of digital technologies and field work. A primary and second bit says locating and accessing a wide range of secondary and using and reorganising data. So what we used to do up here as primary, it's now been moved into secondary. Now, I struggled with that because we have so many models in biology, and I went, well, where are we going to put our pracs with models if it's not a practical? So on page 21 of the syllabus, it includes constructing and using models. So I'm going to include that as a first-hand or practical investigation. All right? So I think you can do the same. I don't know what's happened and why that occurs, but that's okay. All right, so that's the map. Now you might like to do that, get the kids to draw that themselves of the syllabus. I don't know that the syllabus is going to be any use for the kids anymore. You know how we used to give it to them and say, this will help you study. I don't think that's going to help anymore. Maybe a teaching program might. If we have a good teaching program that unpacks each of these points in the syllabus into exactly what you do in order to learn that content statement, that might be the way to go. So it's something that we really should be considering. How are we going to give the kids something to refer back to? Because good old days of nothing is not good enough anymore, really. We've got to give them something. All right, so let's move on and look at cell structure, which is the first part of the syllabus. The inquiry question says, what distinguishes one cell from another cell? We have to keep focusing on inquiry question all the time. So every time we teach content, we say, what was the inquiry question again? If we don't, we will drift. And it's very easy to drift and do other content if we don't focus on it. So what distinguishes one cell from another? First thing we have to do is investigate different cellular structures. Now it says including but not limited to. So what that means is we have to do everything that's listed underneath it, but we can do more than that if we want. Now I think we need to be looking at the more to give more extensive knowledge about an area in biology. So the first one says examining a variety of prokaryotic and eukaryotic cells. So I've just put a little bit of an activity up here now, and I went, well, how could we engage students in looking at a variety of cells and make it interesting? When I first thought of it, I went, oh, well, we'll give them all these lots of images of cells and they can classify them, which is it's what I've still included there. And then I went, well, I love that. I find that interesting, but would the kids? So I went up and I thought, well, what about we look up someone who was around, who was involved in finding the first cell and tell or weave a story. So there's plenty of little stories about von Leeuwenhoek. Find out who he was and then weave a story. This really funny guy that had hair like this, and you know, you could really weave it into a good story if you'd like. And he discovered that if he melted sand and to make glass, he could draw it out into a really fine needle-like shape, and if he heated it again, he could get a little clump, a circle sphere on the end of this piece of glass. And then he discovered that if he put it onto something, it made it look bigger. So he then said, I'm going to look at stuff. So all of a sudden, he can start to see things that before he couldn't see. And he found those little animalcules. What is it? Animalcules. That sounds good to me. And you can go from there. So let's investigate and go through that way to look at then. Well, once we had that technology, we've got a lot better, and now we can see what's inside living things. So then you can give them a range of images of prokaryotic and eukaryotic cells, and perhaps non-living as well, and classify them into groups. The reason I say that is because some of them might still think that water or fire is living. If they haven't got through that in the junior school, that still may be a problem. They need to know that the only thing, the reason they are classed as living is because they're made of liquid cells. So give them these images and ask them to classify them into groups. So they will then have this group of cells that don't have any nucleus in them, a group of cells that do, pretty much that's what will happen. And then you go on and give the details about the different types of cells. So in the Moodle, I've provided you with a table with distinguishing features between eukaryotic and prokaryotic cells. So just have a look at that for a moment. What you need to do is click on, go through the top here, click on cell structures, down bottom here, go to the, but a variety of technologies I thought at least was 4. 2 light, 2 electron. You don't find much about the individual organelles at this stage. It's really just a case of a very generic, general, we can find out the function of the cell this way. Of the organelle or the cell? Well, is the cell dividing? Or well, we're doing that later. That comes up shortly. What we're about to do is to look at cell organelles and their arrangement. So I suppose the purpose of this is to look at technology that can tell us structure and function of cells. So now let's look at some of the organelles and their arrangement inside those cells that we've just found. Now if you want to link it to that, fine. Okay. See what I'm doing an inquiry approach where the students investigate and explore, and from that exploration they would say things like, well, why does a nucleus have a pore in it? What's the purpose of the pore in the nucleus? And why is it that the Golgi bodies are really close to the endoplasmic reticulum? And that's when we go into the, why is that happening? And then we can go and talk about why. So, well, they do know division. They do know, yeah, no, we can't do much more than that. There was one other. Oh yeah, phagocytosis was the other one. So not in junior, engulfing, but they'll see that one cell comes in and engulfs another. So therefore that's giving them information that can happen. So Eva is saying that there's not very many cell functions that we can use the microscopy to find. The 2 that are in the Moodle are cell division and phagocytosis, but the issue is they won't know phagocytosis because that's not in the junior school. So they don't have to actually name it at this stage. It's really just it's a very general, what technology can show us. So if we look at it from perhaps the technology of the confocal can show us that cells split into 2. The technology shows us that one cell can surround and engulf another cell. So without the technology, we would never have seen that. And that's probably the best I can come up with. If you've got another idea, Rella? Perfect. Yes, so, and that would segue perfectly into, okay, let's look at what you can see inside the cell because of the technology. So that's a similar. Yep. we want them to say, what is it called? Oh, here am I not repeating the question. If we can keep your comments shorter, because I've got to paraphrase it. So the idea is that we teach the light microscope and the electron microscope can allow us to see more about the structure of the cells, and then the students can say, well, why is that there? Why is that like the way it is? And then we can go on to the next section, which is about organelles, and we can name them. So then we can name the organelles when we get to it. Okay, that way the students should be able to apply, use, it's like that 5 E's, explore and then explain coming up. Are there any other questions or comments? Do you think that will flow? Any comments from our online people? Missing words thing at the bottom. If you do that now, don't worry if you don't get it fully right, because it's a bit fussy. But if we're going to jump up now to drawing cell diagrams. So the next page is drawing cell scale diagrams, and that's not necessarily new because we already do it in Year 12 with drawing red blood cells. So what I did was I went and I created a document. You can see in blue here. If you look at that link, it's a clear set of steps to follow that you can create a lesson with or do something with if you like. And I had to run this by the mathematicians because I'd fallen into some bad mathematics habits at the time. So how to draw a cell to scale involves firstly estimating the size of the cell, which is what we do now, and then drawing a diagram of a very small cell, and then estimating the real size of an object if they're given a scale. So it's broken up into part A, part B, part C. Yes, we are. Yes, we are. Oh, sorry. Eva said, are we drawing what we see under the microscope? The answer was yes. And are we using a field of view? The answer is yes. Unless you wanted them to draw a cell. So it depends on your instruction. If you're asking them to draw what they see, then it will be, the field of view will have a scale to it. But if you're asking them to draw a cell out of what they've seen today, like on the next page, draw a cell to scale, then it wouldn't have a field of view. So it depends on your instruction. Is that all right? So the first part here is pretty much repeating what we did with red blood cells. So there's nothing new there. Second part is where I got my hand slapped, because over time I've let slip with the units next to the numbers. So it's not always possible to draw on paper the actual size. They may be too small or too large. So if we use 1 centimetre on your drawing to represent 7 micrometers, which is probably what we do now, can write it as 1 centimetre to 7 micrometers, or 1 centimetre to 7 micrometers. But to express the ratio without a unit, you have to convert both to the same unit. So we can't just write 1 to 7. It has to include units. You're all nodding, so I'm really, aren't I? Okay, so like that. That's 1 centimetre. That's 7 micrometers. So that's the way we're doing it now. That's the way we're doing it now. But if you wanted to write that as a ratio, 1 to 7, you can't do 1 to 7 because you have to convert it to this way. So if on the HSC it came out and it was this way, our kids would be in trouble if we had only ever shown them that way, because they may not understand that way. So we need to teach both. Yep. So anyway, there's some examples for you to use, and you can see that's the first one. Then there's another one under that about the tree, and the next one was part C, and this one is about estimating a size, a true size of something from a scale diagram, which they can easily do on the HSC. So I've just gone through and provided you with the numeracy component of how to work that out. So you can use that as a model if you like. And is everyone okay with doing the numeracy of this type of task? And then there's some examples there at the bottom that you might like to use with your students. Alright, so that's probably a lesson in itself, but that's there with drawing a scale diagram. Now one of the aspects one of the things I included with the Moodle is misconceptions that the kids bring with them from junior school, because they draw plant cells as squares or hexagons, and we draw animal cells as circles. So that, to me, we can work with that in junior school when we do cells in year 7, if we can get them out of that habit so that they understand cells come in lots of different shapes. But while they do come in different shapes, there is a generic pattern to that. Alright, so if you go back and have a look at that Moodle, you've got some pedagogy practice there, things that practices that teachers need, and there's a little bit more on numeracy there about converting millimeters, microns and nanometers. They need practice converting millimeters to microns and back again, and converting microns to nanometers and back again. Because we have, the majority of cells are at micron or millimeter level, but then we have viruses at the nanometer scale. So one of the suggestions at that point would be to don't forget to show them one of these diagrams, which we all do anyway, just to remind them of where the cell fits or the crystal fits in the scheme of things. Any other suggestions for that at this stage? We also, when we're drawing our field of view, I just wanted to talk about the difference between a map diagram and a cell diagram. Having marked HSC, I've seen both, so we should probably teach both to the students. So the map diagram just shows a rough area of where something is, and the cell diagram specifically draws the cells within the field of view. And you can do section in detail quite easily. They don't have to fill in the whole circle.
So, Margaret, can you explain that a little bit more in detail? Because I saw some papers that were marked last week, and the kids were actually doing a lot of sketching. They were drawing the longitudinal section showing the phloem and the xylem. But they weren't being marked down for having sketched. What would happen in the HSC with the marking centre?
Okay, if you're drawing a diagram, you can only draw a single line. You can't sketch squiggly lines, you can't do feathery lines, you can't do colouring in. So if we are asking students to draw a diagram, what is it? Why are we asking them to draw the diagram? Is our question, because that will formulate our marking guideline. If all we want them to do is label it, it doesn't matter. But our challenge is to not let that happen. We won't accept poor diagrams just because we only want the labels. So we have to stop and say, well, the purpose of this question is what? Therefore, what might be required in my marking guideline. So I would include the word accurate diagram to ensure that they use the right processes of drawing scientific diagrams or cell diagrams. Does that answer your question? Any other comments about that? Alright, there is no activity at the bottom for that one, so you can move on to the next slide. Now this one was quite complicated, and I rang someone at the board about this and I was not given a very satisfactory answer for this. The question is about cell organelles and their arrangement. So I said to the lesser people, what do you mean by arrangement? So after lots of discussions with a lot of people talking about the chloroplasts will be near the surface of the cell on the leaf, it sort of seems a bit basic, and maybe for some kids that's what we do. But the more I read, the more I realise that we probably need to do the cytoskeleton in more detail, because it's the cytoskeleton that determines where the organelles are. Now, we've not done that before. That's quite new for us, but it's quite interesting. Once we start investigating it, it makes cell biology brilliant, because all of a sudden we're lifting from the stuff that we've done in the junior school. Now we're really explaining why the organelles are where they are and how they stay there. So I've made a few comments there, and I'm more than happy to go through that another time, because it's a little bit complicated to do in this time. But what I'd like to do now is look at a document I created for you, and it's still a document in growing, because it was really hard, I have to say. What we have to do is because we're still under the distinguish between prokaryotic and eukaryotic cells, I thought, well, we still need to identify which cells have which organelles in them. So then we need to decide, well, which structures are in fact organelles or which are not. And I have to say, I think I've taught ribosomes as an organelle, which they're not, because they're only a molecule. I went, ooh, I think I need to do a bit more reading. So ribosomes are not an organelle as such, because it's just a macromolecule. That was interesting. These are the things you learn when you start going looking. Anyway, so then I thought, well, because we're looking at the structure, we need to know the structure and the function of the organelle, and this is where it comes in now. We've seen micrographs with these black things in the middle. They're now called the nucleus. Anything with a black blob in the middle will be the nucleus, and it's there for this reason. And then I've added arrangement on the end. Now this is just content for you at this stage. I haven't got a learning strategy for you about this. This is just the content. What you'll see as you go through it is it's quite substantial. Now, I was very disappointed with the NESA sample program because it didn't go into this at all. It didn't break it up, didn't explain it, so I was none the wiser after reading the sample program. Now, there's an awful lot of organelles there. And it was only when I was doing signaling, cell signaling in membranes, fluid mosaic membrane, protein channels for signaling, I thought, well, then I need to include an organelle in here as well. So I started then, I went, well, we need cytoskeleton because of the fact that it keeps all of the organelles in place, and then we need to do pillar and cilia as well. So stay tuned, there'll be more information in there. What I would like you to be aware of, compare cell organelles and arrangements. So have a quick look at that and see if there's anything in there that you found interesting. I'll attach it to it. There is also a sample comparison document that I'll attach to this later, and it actually gives you some exemplar, 3 different levels, lower, middle and upper level writing for comparing the similarities and differences between the organelles and their arrangements. So I'll attach that for you later to have a look at, and you can use that in class. Alicia? Alicia is suggesting adding images to this table. I don't know whether I'd give this table to the kids in its entirety. Yes, so by putting an image there, that makes it clear for the students. Any other comment? One of the focuses of this section is the movement of substances into, through and out of the cell. So that's why the cytoskeleton is so important, because that holds the structures in place so that when a protein is created, for example in the endoplasmic reticulum, such as the plasma cells produce the antibodies there, they use exocytosis then through the Golgi bodies, producing those little vesicles to the surface to release the antibodies into the external membrane. So our students now are looking at how things are moving through the cell in a lot more detail than before, which I think is quite interesting. It's almost like opening up a new world for them to what we've done in the past. It's very complicated. Yes. This is about the arrangement. So I suppose what I'm saying to you is a philosophy of how all this fits together. I can only give you the information here, but I'm trying to explain that it's not just about single bits of information. It's a whole story of transport through the cell. So that's why the endoplasmic reticulum is attached to the nucleus, so that the so later on, when you do protein synthesis, you know, we can go back to and refer to it in a much clearer picture than we may have, because in the past, I don't know, maybe I'm not very good, but I just said, oh, ribosomes are the protein makers. That's what, full stop, let's move on. You know, and here we're saying, well, no, let's take a step back. Let's say, well, let's spend some more time on that structure of the cell itself and how it's put together and how things move in and out. So I think there's lots of great possibilities with it. But yes, well, one of the findings of our review is that we're not challenging the kids enough. It's true. That's what it's saying, that we don't expect kids can do things when in fact they can. So what does that mean? Does that mean we need to start at year 7, expecting more, but we're not going to wait until the year 7s get to us so we can start expecting more now? Whoa, raise the bar.
If the primary teachers are doing their job, they're already expecting a lot more in the primary school. Sorry? Oh, they are, but they're not changing the science so much, they're adding in the technology. Yeah, they're adding in the digital technology and they're taking some of the technologies out. But yes, it is being revamped, but they're still going to come through with a lot more than what they had in the past.
Which hopefully is the case. The suggestion is that increase the rigour and inquiry in year 7 rather than starting it now in year 11. Well, don't disagree with you. Then you have to go on and do the fluid mosaic model, again, which is not new. But now that I've given you that overview of how things move in and out, it has a different level now than it has done in the past. We've looked at how membranes are selectively permeable, they allow things in and out, but now we're going to look at, in more detail, the structures and the practice of the protein channels. Yes, you can do sodium-potassium pump. And, yeah, so it's bumped up to a new level for here. Now, when I started unpacking this a little bit more, I asked someone, well, when did you teach lipids, carbohydrates and proteins in stage 4 or 5? And the answer was in digestion in stage 4. So better do a little bit of background information there about that before we go into structures of the fluid mosaic. Dale, did you have a question? No? Okay. Yes?
So at the moment in year 11, we have dot points about organic compounds. So we teach the properties of lipids, and then later on we teach phospholipid bilayer, and then we use the properties of lipids to explain why the bilayer works and why it's suitable for that function. Is there anything like that in the new syllabus where we do the organic chemistry and we talk about the structure of a protein, so later on they understand it's built of amino acids and that's why, translation transcription makes sense, or we're not going there?
Good question. In the next section we're going to look at the alpha, beta, tertiary and quaternary structure of proteins. Now not lipids. But you will. I know I did somewhere ago. I need to do in here, I found you a model of a phospholipid. There's a Molymod kit that sells a section on how to put a phospholipid together. Okay? Yeah. So I've given you the link to the Molymod kit, and I've also given you a link to a totally brilliant model of a protein, but we'll come to that later. And it shows you the 2 alpha chains, the single ones, Then it asks you to make them a beta chain by curling and joining bits. Then you add them together, the carbon, hydrogen, nitrogen, oxygen together to form hydrogen bonds to make a tertiary, and then you bend some of the chains to make it the quaternary, and then all of a sudden you have a beautiful protein. So, yes. Oh, it's on the computer. It's not. Monomods you can order. That one's just a cut and paste paper one. Maybe when we...
I've had 2 meetings with the lab techs this year, and I've introduced them in the inner west and the eastern region to the new syllabus, and we've had a look at a couple of the first units for next year, and I've said to them, talk to your people at the last month of the year and try and get from them a list of what you people need to have for next year. We don't know the answer yet. We're only unpacking it ourselves. But don't be surprised if your lab techs say, what do you guys need for next year? So be prepared. Jot these things down as you go. And even the lab tech said to me, for prokaryotic and eukaryotic cells, the best stuff is the slide viewers. What do you call the bio viewers? They said they're really great on that. And if you haven't got the prokaryotic, eukaryotic stuff on your bio viewers, maybe you might even like to buy them. So I think this is where you've got to have a lot of dialogue with your lab techs and start prime them up.
And you can't see, that's it, you can't see them. That's right, yeah. You can see protozoa, so that might be a way to go. No. Protozoa? Yeah. We're doing it on the prac day. We're going to do all these pracs on the prac day, so we're working at the moment on a list of equipment for that. So once we've done that, we might share that with you. But you will have to look at your slide box and do a bit of a not chuck out, just move them into another box to come back in 10 years' time, the current one, to replace and renew some. And the blood slides obviously will stay. We'll use those still. So given that we're looking at the transport of materials into and out of a cell, we need to make sure the kids know about glycolipids, glycoproteins, what the transmembrane proteins are, which all links back down to the cytoskeleton as well, because the cytoskeleton is made up of, I think, there's all the information for it. The cytoskeleton is made up of protein, of which there are 3 classes, and you can see some of them are part of the membrane structure itself, and some of them, the tubules, are what holds the parts inside the cell. So I've included that as a mind map for you, because that was new for me and I hadn't done that before. Now what are we going to do for the bottom kids with all these new words? Look, it's about how we teach it and how we repeat the same words over and over and over until they hear it to death and then they get it. Lightning writing as well. Alright, so again, because we're doing distinguishing between, there's something about the membranes. They contain carbohydrates and sterols in eukaryotic cells, but not in the prokaryotic cells. So now we're moving to the next section, which we should have been at ages ago, but that's okay, on cell function.